rotation of sigma bonds, and we were talking about the Newman projection looking down two sp3 hybridized carbon atoms. And then if we use these blue and red balls of different sort of markers, how staggered through eclipse. As you rotate through the eclipse, you experience strain. So we'll use that word quite a bit. Strain meaning higher energy. And if something's higher energy, that will basically influence the reactivity. So later on, more towards module 5, really, this will come back to haunt us in a way. So again, as we learn things, keep in mind we're not flushing them down the toilet. They will come in handy later. So that would be the first half of the lecture. In the second half, we're going to get to cyclic conformational analysis. So you count sort of the carbons, so I'm holding a carbon. This ring, cyclohexane, the 6-membered ring, is especially important in nature and organic chemistry. And we'll talk about why that is in relation to strain. So we'll do a little bit of math, nothing terribly complicated, sort of normalizing strain from acyclic to cyclic systems. And then as you see on this ring, we have two markers, a purple and a red spongy ball here. And so what we're going to do is something like that. Cheap magic trick. You'll see that the difference in orientation of if you look at the purple one, it's sort of up, and now it's not up anymore. It's subtle, but that makes a difference when we get to the cyclic systems. So that will be our lecture today, and then on Friday, we're going to move into another set of isomers called stereoisomers. And so you've sort of seen that already throughout the semester, but we're really going to sort of quantify it in terms of nomenclature. So you know the nomenclature rules. We're going to add another layer to the lasagna and make it a smidge more complicated, but not too much. You do have your model kits. You probably want to bring those on Friday, because so the thing we're going to talk about are stereoisomers. Those are mirror images. So it really helps to have your model kit so you can build those and see the concept we're talking about. Any questions before we begin? So we had left off with propane, and then we got cut off with butane here, so that's what we'll start with. So the first set of questions is specifically related to butane, in that what we'll see is when we start plotting the strain energies, the curve looks a little different than ethane and propane do. And again, that's based on analyzing, I want to say 4 discrete conformers. Keep in mind that, again, it's very subtle, but that's kind of what organic chemistry is. It's all about things being subtle. So here we have a conformer. If we consider the red and blue spheres as methyl groups, then this would be butane. And so even if I tilt it a little bit, that's considered a different conformer. But really, we're just considering staggered and eclipse for the purposes of this class. So, but any little rotation makes it different. And so let's draw butane out. We'll walk through this exercise so we're all calibrated about how to approach these problems. So butane, we know is C4H10. So named molecular formula, it's a linear hydrocarbon. We'll go ahead and draw it out using just the line drawing. I'll throw some numbers in there. So we've numbered it so we can approach what this problem is asking. We want to look down C2 to C3. So C2 being the front carbon of the Newman projection and C3 being the back carbon. And let me think about this. So I want to draw the front carbon like that. And I'll put in the circle for the back carbon. And then the way this is drawn, the substituents coming off the back carbon are all staggered right now. So up, left, right. And our eyeball is right here. And we're looking down 2 to 3. So for our purposes in organic 1, we're really only going to look down sigma bonds. And remember, they freely rotate. So hopefully this is clear. If not, I'll grab the model kit. But how do I want to explain this? So we have carbon 2. That's this front carbon right there. We have to put 2 hydrogens and a methyl group on there. So where should the methyl group go? On the bottom? Is that clear to everyone by looking at sort of the flat drawing? So then the left and the right there have to be H's. And then on carbon 3, where should the methyl group go? On the top, so up. And I guess what I'm getting at, when you're looking at that, see how that bond is sort of angled down? And then this bond is angled up. So you can use that to orient yourself in doing this. And one of the things when we're talking about these is it's sort of easiest to calibrate yourself to 2 substituents. So we're looking down C2 to C3, and then what we'll talk about is the methyl groups, which are 1 and 4. So right now, the dihedral angle between the methyl groups is what?
Can you say that again?
The dihedral angle between this carbon 1 and carbon 4 is what?
180.
180 degrees. So again, dihedral angle involves 4 atoms. And so what we're talking about there is C1, C2, C3, and C4. So that's a staggered conformation. And if we're specifically referring to both methyl groups, we would also call that the anti-conformer. So anti meaning opposed. And now what we're going to do, how do I want to do this? Maybe I'll rotate the front carbon atom. So we'll leave the back carbon atom as it's drawn, and then we'll just do these 60-degree rotations. Or wait, 120-degree rotations. 60? 60, is that right? Okay, 60. So now when we do that rotation, the conformation is go from staggered to eclipsed. So how can I draw this the prettiest I can? So there's the front, there's the back carbon, and then I'll just draw sort of parallel bonds from the back. So the back carbon stayed the same. That methyl group now moved over clockwise. So CH3, H, and H. So that's now eclipsed. That's going to be higher in energy. And the next 4 slides, we'll go through the calculation of what the strain is for those eclipsings. So there's 3 eclipsings and then this other one called gauche. So far we have 2. We can eliminate choice A. Now we'll do another rotation to make it staggered again. So even though that's staggered, it does experience some strain. So typically what you'll see in like textbooks is like a little ball or something. And that's called steric strain. So as we learn these phenomenon in module 3 here, we'll start talking about sterics and electronics. Electronics have a little bit more to do with like things like resonance and electronegativity. Steric you can think of as like bulk. And again, the reason this model isn't the most accurate. So this is a ball and stick model, but again, the atoms have a van der Waals radii, so they're much larger than they sort of appear in this model. And when they're in this gauche interaction, these are methyl groups. The hydrogens coming off that, they sort of feel one another, so they want to move away in that strain. So as these things rotate from gauche and then the next one eclipsed, that strain is going to get larger because they're pushing each other apart. And what that does is distorts the bond angle. So again, things want to be happy in their sort of average environment. So this is gauche when we're talking about the methyls. And then we do another rotation, and we'll see now that the methyl groups are going to be eclipsed. So as you do this, as I said, we're talking about discrete conformers, not sort of the minute rotations. When you go through this exercise, you're going staggered, eclipsed. They just alternate. And now, what is the dihedral angle between those 2 methyl groups? So the dihedral angle here is 0, and this is called syn. So SYN. And the syn and anti-conformers, when we move into module 5, are going to be the most important for us when we learn about a reaction mechanism called elimination. So if you do the next rotation clockwise, what you'll find is that you end up with another gauche conformer. So overall, we end up with four unique conformers. So on the next slide, we'll look at the energy penalties and calculate that for each. And then the 5th slide will show the plot. So we have a staggered conformer. The dihedral angle of the methyl groups, that's 180. So we call that anti. So there's really no steric strain involved in this conformer. If we look at, this table here of energy penalties, we set it staggered. The methyl groups are not gauche, so we say this has a relative energy of 0 kilojoules per mole. So on this one, we did a rotation. Now they're eclipsed. And so basically, we have 3 eclipsing interactions. We go to the table, identify what they are, and then add the sum. Question? A. So this is what you're looking for. We'll just start here. So you have HH eclipsing. So we go over to the table, and we just write that to keep track of the math. So that's 4 kilojoules per mole. Then we have methyl H. That's going to be plus 6. And then another methyl H, that's plus 6. So we add that up. We get 10 plus 6, 16. So C is our answer. So we'll rotate again. Now we're staggered, but we see this relationship here between the 2 methyls. That's the gauche. There's only one of them, so that's going to be 3.8 or B. One thing I want to point out, these are essentially the 4 numbers you need to know. If say you're looking at a molecule, but this is not CH3, but like CH2, and then it's bonded to some other stuff, you just approximate that whole other thing there as methyl. So if this was CH2, CH3, you'd say, yeah, that's a gauche interaction, and it's 3.8. So that's kind of an engineering approach to a first approximation of things. And then if you need to refine to a second approximation, there's probably other tables you can find data with. And then the last eclipsed, where we have the syn relationship between the methyls. We have plus 4, and then the big one here now is plus 11. So that's 15 plus 4, that's 19, so the answer is D. So we have those 4 data points which we can use that template to plot the conformers on top. We rotate through the dihedral angles. Oh, there's kind of a mistake here in terms of that dihedral angle matching with what that conformer is in terms of the name. But anyway, the data is still plotted correct. So for staggered anti, that's 0 kilojoules per mole. Then you go through the rotations. You have 16, goes down to the gauche 3.8, up to the highest 19. And then overall, the symmetry of this plot lies right here. So you can see that as you start going back through those rotations, the rest of this plot will look exactly how
does here. One further thing to point out. So that would be the strain maximum. That's the highest. And then you're staggered with anti, that's the strain minimum. Within those 2 points, you also have what's called local. So this would be a local maximum. And then this point here would be a local minimum. So what's, if you were to go further in organic chemistry, which maybe one or two of you might, that would warm my heart tremendously, but really the way this looks, it's you know, here you see a two-dimensional plot, but in reality, the way these things exist, it's like a 3D sort of mountain range. If you can imagine, like those humps are like hills, that's how it actually looks. So later on, when we talk about mechanisms and energies, you know, you're going up a hill, you hit the top. If you existed as a ball, when you get to the top, you roll back down sort of immediately because that's the highest energy. But on this plot or roller coaster, you go this way or that way. But in reality, you could sort of go forward or backwards off that as well. So this is like a slice of the mountain range, really. So again, the best way to think about it is a mountain range that you're sort of hiking on. But keep in mind, you know, if you are doing work, it takes work to get up there, that strain. If you're sliding down, you're getting to these minimum points. But keep in mind local and global in terms of maximum and minimum, because those are the conformers that we'll study later on. So on your own, what you can do to practice this, just look at a couple of these compounds. Pay attention to how this is worded, what bond you're looking down. Do the same activity, plot the points, and see how those plots look. I made a couple of short videos like 2 or 3 years ago that actually shows these rotations through a computer program. So you see the rotation and then you see the plot of the energy moving along. So you can see as it hits that maximum, what does that conformer look like? So these may be the ones that I did in the videos, I do not remember. So any questions on acyclic? Yes. So as again, part of your skills now, you've learned nomenclature. How do you take a name, convert it to the structure, and then do these other type of activities? So in converting a name to a structure, start with the parent, so butane. And then number the chain. And then you're going to get to the rest of the molecule. So positions 2 and 3 each have a methyl group. And so that would be 2,3 dimethylbutane. And just to get you started here, if we look down the C2 to C3 bond, so keep in mind that carbon 2 is the front carbon. So now that's the front carbon. That's going to have 2 methyl groups coming off it and then a hydrogen. Carbon 3 also has 2 methyl groups and a hydrogen. So that would be a Newman projection conformer of that. Then when you go through those rotations, you'll see it has different eclipsing interactions, which are going to lead to higher energies that you would then plot and sort of connect the dots. Yeah.
So how do you know where to stick like the CH3s and the H's? Like how do you know where like if this one goes right to the top and this one goes?
So the question was, how do I know where to draw like these CH3 groups? So this one here, I drew down. That one right there, I drew up. Because it sort of follows that. We call that a zigzag. But these other two, say here and here, for now, it doesn't matter where you draw them. But to give you a preview of when it will matter, let me so if I drew that molecule, so notice this bond I put like bolded, we call it a wedge. So then it would matter because that's going to relate to what we start learning on Friday in terms of stereochemistry. So let me just put in the other substituent there so it's a little clearer. We show 3 bonds, so the last thing is hydrogen. So if we were looking down that carbon bond there, right here, now it would matter where we put those things because we've specified that this group, which is coming out, which one of these, A, B, or C, where should I draw this group? On position B. So C would be the methyl group that's down, and then A would be the hydrogen. So again, as we learn a little bit more on Friday, we merge all this stuff and sort of build upon it. The reason it doesn't matter here Did something weird there. The reason it doesn't matter there, see that carbon atom? That carbon has two of the same substituent. This carbon here has four different substituents: a methyl group, an ethyl group, and a butyl group, and the hydrogen. So that's a preview of Friday. When a carbon atom has four different things bonded to it, it's a stereocenter. Any further questions? So in moving from alkanes to now hydrocarbons with rings, specifically cycloalkanes, we've determined strain energy through the Newman projection and doing the rotations. But we benchmark linear hydrocarbons or acyclic hydrocarbons. Outside of any eclipsing interactions, we benchmark them to have zero strain. And so this is relative to understanding cyclic alkanes. So we know that an sp3 hybridized carbon has a bond angle of 109.5, meaning that that's the average bond angle. There's no compression or expansion of that. And so in relation to this equation here, we're going to quantify strain in ring systems based on combustion. So combustion is when you take a material, as you see this molecular formula here, that's for a cyclic alkane. If you take 3/2 mole percent of oxygen, you add a spark, you form N moles of carbon dioxide, N moles of water, and N moles of heat. And experimentally, that's what you measure. So it's like calorimetry, which you learned in Gen Chem. Heat is the thing that we're measuring. And so based on the amount of heat produced from combustion, you can determine the strain energy present in a molecule. So linear hydrocarbons, we just say there's sort of no angle strain. But then when we get to the cyclic ones, as we'll see in the table, there is strain present, it decreases to zero, and then it starts increasing again. And so on this table, if we look at ring size, increasing from 3 to 8. I've highlighted 6 in green, cyclohexane, because that's the within that boundary of sort of ring sizes, the one where the bond angle of the carbon matches the normal sp3 hybridization. And so basically, there's no angle strain. This value here, 3951, that's the heat that's measured in kilojoules per mole after you combust it. If you take that and divide by this number here, 6, you get 658.6. So overall, that's set to 0. So the 6-membered ring has no strain. So in a sense, it's like a linear hydrocarbon. Now, if we circle back to a 3-membered ring, cyclopropane, So I mean, even drawing it, you can see that those bond angles do not look like 109.5. So you've taken a normal bond angle and you've compressed it. And so anyone do the pec deck at the gym? You gym bros and gals out there, you go like this, and you're squeezing it, right? That's strain. So if you're taking something normal and you're squeezing it, you're adding energy. And so that's what's happening with cyclopropane. You're putting a lot of energy into that molecule relative to the CH2 groups present. So CH2, 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 all of these are sp3 hybridized. So the bond angle is 60. So you've compressed it about negative 50 degrees from what it wants to be. When you combust it with oxygen, that's the value you get, 2091. If you take that and divide it by 3, you get 697. And so the difference between these here, 697 and 658 is 38.5. So when you multiply that by 3, you get 115. So this column normalizes it per CH2 group present in the ring. And then the last column just multiplies it by the number of carbons. So cyclopropane has a strain energy of 115 kilojoules per mole. And practically speaking, what does that mean? It wants to react with something to open that ring up and relieve that energy. So a lot of polymer science is based on using small membered rings because as a monomer you can open it up to form something that's a reactive intermediate that will react with another monomer, and eventually you grow out your polymer chain. So any questions on that logic? So again, the combustion experiment is what we do to produce something we can measure, and that measurement is heat, which is through math and equations calculated back into energy. So 5 and 6, cyclobutane, cyclopentane, you can see that the angle is definitely approaching what it should be at 109.5. So the compression decreases. The energy is measured through combustion. You normalize it and see in the last column it's essentially decreasing. So as this number decreases, the strain present in the ring decreases. It gets to 0 at cyclohexane, which essentially there's no ring strain. And that's the inflection point where you get to 7 and 8 membered rings. It starts increasing again. Even though these bond angles are still normal to 109.5, other strain factors start popping in, and we'll give a preview of that today. So that's a very cheap explanation of a 6-membered ring, but it's very important in organic chemistry. It's important in nature because in nature, a lot of things are present in 6-membered rings. If you go on to take organic 2 with me in the spring, we'll see that glucose, so a food source that what we use as humans, glucose essentially exists in a 6-membered ring. And then from that, you can form the 5-membered rings like you see in DNA and RNA. But the 6-membered ring is preferred because it has the least amount of strain. So because it's so prevalent, we're going to talk the last 20 minutes here about it. And there you see it. It's a 6-membered ring. I have it tattooed all over my forearm. I like it so much. But when you see it flat, you're like, okay, there's 6 CH2 groups. But that's really not the best representation because the hydrogens in that molecule are not all equivalent. Even though if we were doing the uniqueness, we would say, yes, there's one set of 12 unique, or 12 of the same type. But the molecule doesn't sit flat. And as I mentioned in the prior lecture, Barton and Hassel won a Nobel Prize for really translating the sort of flatness into a 3D topology. And from that, what they learned is that the hydrogens are actually different. So what you see here in these drawings are called chairs. So it's you can kind of imagine, you're sitting in the chair, eating your Doritos and drinking your Mountain Dew, watching football. So there's the chair form, and then there's another thing called a boat form, which we'll see in a little bit. And so just like with the Newman projections, when you were doing rotations, you had staggered and eclipsed. For cyclohexanes, you have chairs and boats. Any SNL fans? Anybody on a boat out there? Nobody? Okay. So in their time, it's kind of hard to see, but that letter there says P. That means polar. And this letter here is E. That's equatorial. Nowadays, we don't use polar. We call it axial. So a cyclohexane ring has 2 types of hydrogen, axial and equatorial. So if you count them, you have 1, 2, 3, 4, 5, 6 axial, 1, 2, 3, 4, 5, 6 equatorial. In the bottom 2 structures, what you'll notice, see this thing here, it's called R. R is like the X variable in algebra. It's whatever else is in the molecule. The R there is equatorial. The R here is polar. The difference between these 2 is called a chair flip. And so one of those conformers is more stable than the other, and we'll use the model kit to help us understand which one that is and how do they convert from one to the other. So to put this in perspective, why is it important? Here's a molecule called cholesterol. You've all heard of it. Generally, it has like negative connotations if you watch the news and health reports, high cholesterol, blah, blah, blah. So you get it from food, but it's also made in your body. And so cholesterol is a modified steroid. And so what is a steroid? Steroids typically have 4 rings, A, B, C, and D. And you can see in there that A, B, and C are all 6-membered rings, right? And then the 5th ring, D, is 5-membered. But it's made in your body every day. Look at that, that's pretty amazing. Over 37 chemical steps. And these are not like you in lab on Tuesday from 7 to 9 p.m. spending 3 hours. I mean, these reactions happen very fast. And cholesterol is a structural component of cell membranes. So if you're into biology and you study how a cell membrane has the lipid bilayer, cholesterol embeds itself in that, and then it's released when it's needed. So it's a starting material for other steroid hormones such as bile acid and vitamin D. And a hormone is something that in your body triggers another biochemical process to start, continue, or stop. So here is the name. Aren't you glad you don't have to name that? So we just call it cholesterol. But the name, again, if you apply the IUPAC rules, that's the name you would get. And so there's the flat, and then this is the 3D version of it. So we're not going to get anything too complex like that. What we'll spend our time doing is taking just a flat cyclohexane and drawing it into a chair. And then looking at substituents like this alcohol here. How do we know to draw that alcohol in the equatorial position based on the flat drawing? So that's the skill we're going to try to develop. So my graduate advisor said, when you're sitting in seminar, if you're bored, draw chairs. So if you're bored, you can draw chairs. So how do you draw a chair? Every bond in a chair is parallel, so that's sort of your guiding light here. And I'll draw it in red alongside what's drawn there. So parallel lines, they have to be a little bit offset. And so we've drawn like a sort of a V and then the inverse of a V, whatever that might be. And then basically you connect the last two bonds. So parallel, parallel. And then as you see in the line below, the relationship between these two, that's a chair flip. So a lot of, try to keep your bond angles nice and pretty. Organic chemistry is science, but there's a little bit of art to it. If you practice it a while, you get pretty good at it. Does kind of look like a modified Budweiser sign, if you're into that, or Natty Light, whatever. So again, just keep in mind the opposing bonds must be parallel. And so once you have that, how do you stick the hydrogens in there to know if they're axial or equatorial? So let me draw a chair. And we'll walk through the different sides here. So what I'm saying, if your eyeball is up here, I'm going to say that's the top. So see on the top you have 1, 2, 3 carbons pointing up. So the axial hydrogens are just drawn straight up from there. So there's 3 axial on the top. Then you have on the bottom 1, 2, 3 carbons pointing down. So those axial bonds are straight down. So of the 6 axial, you have 3 on top, 3 on bottom. So for the equatorial now, we're going to split the molecule sort of in half. Maybe I'll draw a blue line. And then we have the left half and the right half. So there's 3 equatorial on the left, 3 equatorial on the right. And these are probably the trickiest ones for students to get right. Because again, you don't want to have like a weird bond angle. Like if I bent my arm like the other way, you'd say, man, that's painful. So if you draw a hydrogen that's facing the wrong way, it's again, this sort of painful response. So let's start here. And I need to draw an equatorial hydrogen. But again, the bond needs to be parallel to something. So it's going to be parallel to this existing bond right there. And so overall, you make sort of an N shape. Now, if just for explanation's sake, I'm going to draw in the axial hydrogen on that carbon. Is that axial hydrogen up or down? It's down. So the equatorial hydrogen is what? Well, we would say it's up in terms of like the I don't know, face, for lack of a better way of saying it. Since that's down, that has to be up. Now if we go here, we know that that axial hydrogen is up. So this equatorial hydrogen has to be down. It's going to be parallel to that bond. So again, these things rotate as they go through, up, down, up, down. So the next equatorial one has to be up. And what I mean by like sort of drawing a equatorial hydrogen weird, like if you drew it like that way, that would be like farting in church. People would disrespect you. Or if you did this, if you drew it like in the ring, that would be meh. So you want to sort of obey the bond angles that you know. And then just continuing on with the right. Since that one's up, this one has to be down. Then up, then down. So it may not seem like we've differentiated anything, but once we start substituting hydrogens for other stuff, this will really matter quite a bit. So no bonds pointing into the ring, no weird bond angles, just so you understand, again, how that impacts reactivity at those carbon atoms. So maybe what I'll do here is use the model for a couple minutes, and then we'll probably wrap up on this slide for today. But if you pay attention to the colors, the blues and the red hydrogens from the left to the right structures, you see the blue hydrogens are axials. They go through that boat, and then they go to the other chair. Now the blue hydrogens are equatorial.
the red equatorial ones become the blue equatorial ones over there for the chair flip. So imagine that we've just, the yellows are hydrogens, but these are hydrogens as well. They're just colored so we can see them. So in a chair flip, what's going to happen, in my right hand I have this carbon up, in my left hand is carbon down. So it's going to go first through a boat, which I'm going to flip my left hand like this, and then watch how these things change. So there's the boat. The reason the boat is higher energy is because those things smack into each other. And now with my right hand, I just flip that down. And so this one has now become axial. This is equatorial. So that's what a chair flip is. You have a carbon going down, a carbon going up. And it will really matter when we start considering these things as not hydrogen. So, for example, if I move this over, so see how these are both axial now? So when I do the flip, they both become equatorial. And so I was way too ambitious with the number of slides today, as you can see. But what we'll continue on with Friday is looking at now changing out hydrogen to methyl, doing these flips, and then determining the relationship between them.